Hi everyone, welcome to Ascendia Biology, where you learn it, understand it, and pass it. The last biology channel you will ever need. My name is Adedapo Adeshola Emmanuel. Do you know what WAEC wants from you in biology practical in 2026? Keep watching to find out. Thank you. All right, without much ado, we want to quickly move into what we have in stock today. And this would be the pre-practical session for WAEC 2026. And here we will be looking at some of the questions that are likely to be asked. I will use my 17 years of teaching experience to explain and make you understand some of the things or some of the technicalities that are embedded in WAEC questions. So for today, we'll be looking at the first specimen, which is groundnut pod containing seed. The first likely question that WAEC can ask is identify specimen A, the type of fruit, and give two reasons for your answer. So the answer here is specimen A, groundnut pod containing seeds. Then the type, what is the type of fruit that A belongs to? A is a legume. A is a legume. So what is the reason for identifying A? What is the reason for identifying A or classifying it as a legume? Number one is the presence of pod, and presence of enclosed seed. So if you open it, you are going to find out that seeds are embedded inside the pod. So another question that WAEC can ask is, what is the scientific name for specimen A? What is the scientific name for specimen A? The scientific name for specimen A is Arachis hypogaea. Arachis hypogaea. Some of the mistakes that students make during WAEC is for them not to understand how to write scientific names. While if you want to write scientific names, some of you would start with small letter and then you finish with capital letter, or some of you would write everything in capital letter or everything in small letter, and even if you write it correctly, you are going to underline everything together. This is a mistake. Do not do this. So if you want to write scientific names according to Carolus Linnaeus under the binomial system of nomenclature, the first name starts with the genus and it is written in capital letter, while the second name is the specific name and it is written in small letter. It is written in small letters, and so you underline them separately. If you underline them together, WAEC will not forgive you. So this is one of the tactics, or this is one of the ways that students lose mark. So this one will be a bonus, and this will be just a mark. Another clever question is classify specimen A into kingdom, division, or phylum, and class. So let me quickly explain this. The old system of classification is as shown, where we have kingdom, Plantae, we have division, Tracheophyta, and then we have subdivision, that is Spermatophyta. Because under division Tracheophyta, we have two subdivisions where we have the Pteridophyta and Spermatophyta. The Pteridophyta are non-flowering and they are non-fruit producers. So they do not produce flower and they do not produce fruit. They have other means, most especially through sporulation and other means of reproduction. For the second one is Spermatophyta. The word sperm actually means seed. So that's why they are seed producers and they are flowering plants. And then we have class Angiospermae. Angiospermae. For seed producers, we have those that their seeds are naked, their fruit is born on a special structure known as cone. And then we have the subdivision as dicotyledonate. That is the classification according to the seed leaf. However, this might be a little bit too much for some students. And so what you are to write is for you to be on the safer side, write kingdom, plantae, division, tracheophyta, class, angiospermae, and then you bracket dicotyledonate. The next question under specimen A might be, what type of fruit is specimen A? So I've already answered that in the first question. So I'm repeating it. So the reason it is a legume is that it is a simple fruit, and under simple fruit, it is a dry dehiscent, which means that it has a pod and it has a line of split. Because they are formed from a monocarpous ovary, they are dry dehiscent. They are dry dehiscent. So, and it is under the family of legumes. So that is why it is like that. So give two reasons for your answer. So number one is the presence of pod, and then it develops from a single carpel. It develops from a single carpel, that is a monocarpous ovary, and it belongs to the legume fruit type. So that's what I've just explained. However, legumes do not split on their own. Now, number 6, differentiate between specimen A, specimen K, which is maize grain, and specimen M, which is tomato fruit. If you look at these three, WAEC usually ask questions on fruits. Let's look at some of the answers. So if you look at it, the bean pod specimen A, specimen K is the maize grain, and tomato fruit is specimen M. So what are the differences? So whenever you are asked to differentiate, it is expected that you write your answer in a tabular form. Write your answer in a tabular form. So the first difference is that for specimen A, there is presence of pod, while the two others, K and M, have absence of pod. So when you are comparing, you must compare accordingly. If you are describing the head, don't say one has head and the other one has leg. No. So WAEC will not mark you correctly for that. We will rather say specimen A has pod, specimen K has no pod, specimen M has no pod. So the second one is the class or the type. So the type of fruit for specimen A is legume. It is a legume. While maize is a Caryopsis. Why is it a Caryopsis? Because the pericarp is fused directly with the seed coat. So that is why it is what? It is Caryopsis. It is also a simple fruit. It is also a simple fruit. And the third one is tomato fruit. It is a berry. Why? Because it has many seeds. It has many seeds. Then the next one is the texture. So the pod is dry. So we say it has dry pod. Then for maize, it is dry. So dry maize grain. And then for tomato, it is what? Succulent. It is succulent. Pod as elongated and cylindrical, while maize grain is small and triangular shaped. For tomato, it is big and round. As for the color, so for the bean pod, we have light brown. For maize, it can be white or yellow. And for tomato, it can be red or orange. The next question, list 5 economic importance of specimen A. Number 1, it is a source of protein food. Some of you will wonder that groundnut is more of oil, but it contains protein. So it is a source of protein food. Number 2 is it is also a source of edible oil. That's why we have the groundnut oil. Then it is a source of income. So for those of the people who are into farming, so groundnut is also a source of income. Then number 4 is that it can be used to make peanut butter. And number 5, it can also be used for feeding animals. That is, the pod can be used to feed animals. What is the mode of dispersal of specimen A and K? What is the mode of dispersal of specimen A and K? And I would also want to include M. Mode of dispersal. The mode of dispersal for A, K, and M is animal. Then number 9, state 4 materials necessary for germination of specimen A. So we have suitable soil, then we have water, we have air, and we have viable seed. Sunlight is general, so it's not a major factor. So, but the necessary factors are suitable soil, water, air, and viable seed. So if you put sunlight, you might also be given. Number 9, state observable differences. Between specimen A and B. Specimen A is groundnut pod, while specimen B is roasted or crushed groundnut seed. So let us look at the differences or the observable differences. So first one is presence of pod. The second, specimen B has absence of pod. Then number 2 is that specimen A contains viable seed and it is raw and uncooked, while the other one is cooked because it has been roasted, or it is not viable, unviable seed, so it has been crushed. Then number 3 is the texture, where specimen A is hard but specimen B is soft or powdery. Then number 10, name one nutritional value of specimen A. So we have fat and oil, and fat and oil are also referred to as lipids. So number 11, state 5 nutritional importance of fat and oil or lipids. Number 1 is that it is a source of energy. In fact, research told us that fat and oil, they are a large source. They produce larger amount of energy than carbohydrates. Number 2 is the production of heat and insulation. Number 3 is the protection of organs. And number 4 is food storage. Now we are moving on to specimen B. So what are the questions that are likely to be asked for specimen B? What are the likely questions? They will say, identify specimen B without reason. So specimen B is roasted or crushed groundnut. So it depends on what you are given. So if you are given a crushed groundnut, then you write crushed groundnut. If you are given roasted groundnut seed, then you write roasted groundnut seed. On no account must you write the two together. Do not write roasted or crushed groundnut seed, because these are some of the mistakes that students make during WAEC examination, during biology practical examinations. Number 2, rub a small portion of specimen B on a filter paper. Record your observation and inference. Whenever you see something like this, the first thing that comes to your mind is that you are running a test, and this is a food test. It usually comes out. This is one of the questions that usually comes out. So whenever you are answering this type of question, you are to create a table, and with 3 columns. You have the first column is the test, the second column is the observation, and the third column is the inference. So you are going to write specimen B. You are not going to mention it, and you can also mention it. WAEC will not penalize you for that. So specimen B, or depending on what you are given. So I would rather go with specimen B on filter paper. Specimen B on filter paper. So what will be your observation? The filter paper spot became transparent. It became transparent under what? Under light. Under light. So the inference here is that fat and oil is present, or fat and oil are present, depending on what you want to say. So here you can also say that instead of fat and oil, you can also say lipid is present. I would suggest that you keep to fat and oil. Number 3, name the text performed above. Name the text performed above. So that the answer here is translucent text. Translucent text. Number 4, name other 2 tests for this food substance. The first test that you can perform is the emulsion test, which uses alcohol and water, and a white milky precipitate is observed. Then the second test is Sudan 3 oxide test, where you are going to have a black precipitate. A black precipitate. So a red precipitate will precede the black precipitate. So that is what you have there. Okay, so number 10, place specimen B or A. Specimen B or A is talking about the groundnut seed or the raw groundnut. Either way, they can ask you any one of these. Or specimen J. Or specimen J. Specimen J is fish. Fish. Specimen J is fish. So into specimen C. Specimen C is beaker containing water solution, and D is beaker containing salt solution. So D, for 5 minutes, record your observation and inference. Record your observation and inference, just like we have done for the other questions. We have our test, observation, and inference. So whenever you want to do this, so specimen B or J, that's the first row. The first row is specimen B or specimen A or specimen J into specimen C for 5 minutes. So what you are going to have there is the seed looks slightly larger, or the fish will begin to swell up. If it is the fish that you have put in, the fish will begin to do what? To swell up. So which means that water has moved inside the seed, or water has moved inside the fish. So what has taken place? Your inference will be osmosis has taken place. Then the second row is that you can put specimen B or A or specimen J into specimen D. Specimen D is a beaker containing salt solution. And so what will happen is that the fish looks shrunken or smaller in size. If it is the granite will also shrink. That is if it is specimen A, it will also shrink. So what you are going to have is that plasmolysis has taken place. Another question is, give a name to the 2 phenomena above. Some of us we don't understand the concept of osmosis and diffusion. So diffusion is different from osmosis. In diffusion, it is the solute that moves in the medium, or it moves through the solvent. But in osmosis, it is the solvent that moves through the solute. So in this case, it is the water molecule, the movement of water molecule. We have osmosis. So the first one is that when water moves from the environment into a cell, then we say that that is what? Endosmosis. And an example of that is turgidity. An example of that is turgidity. So turgidity is another form, or a specialized form, of osmosis, while exosmosis is when water moves out of a cell into the environment. That is what? Exosmosis. Exosmosis. So the answer there is osmosis. So now they now said another question might come out. They might say, define the following: osmosis, turgidity, and plasmolysis. Osmosis, turgidity, and plasmolysis. So the first one here is that osmosis is the movement of molecules. Those are keywords. Those are keywords that you need to pay attention to. The movement of molecules from the region of lower concentration to the region of higher concentration through a semi-permeable membrane. Some definitions would define osmosis in terms of water or salt. So if it is water, if it is in terms of water, you will say from the region of higher concentration of water to the region of lower concentration of water. If it is salt, you will say from the region of lower concentration of salt to the region of higher concentration of salt. So the general assumption is that water is hypotonic while salt is hypertonic. So in this case, it is the movement of molecules from the region of lower concentration to the region of higher concentration through a semi-permeable membrane. So the second one here is turgidity. So turgidity is the movement of water molecule into the cell until it becomes rigid. That is, the cell can no longer hold water again. When that cell is placed in hypotonic solution, and that is why water is hypotonic. So it is the movement of water molecule into the cell until it becomes rigid or turgid. And then when it is placed in hypotonic solution. Then the last one is plasmolysis. So plasmolysis can be defined in two ways. So the first one is that it is the shrinking of the cytoplasm when a cell is placed in hypotonic solution. And then the second one is it is the outward movement of water when a cell is placed in hypertonic solution. So those are the concepts, those are the things that you need to pay attention to. So we move on to the next specimen. So specimen E is beaker filled with sandy soil. Number one, identify specimen E and give three reasons for your answer. So specimen E, a beaker filled with sandy soil. What are the reasons for your answer? Number one is presence of grit. Number two, presence of visible pores. Number three, presence of large stones. Number two, second question, in a beaker or conical flask, fold a filter paper into a funnel and put small portion of specimen E. Pour the whole volume of specimen C, which is the water solution, into it. Record the observation and inference. Just like what we have been doing before, you have your test, observation, and inference. So specimen E plus specimen C, that is water. So what is your observation? The same volume of water was filtered through E into the what? Into the conical flask. So what is your inference? So your inference is that there is what? Low or poor water retention capacity. So the third question here says, what is the name of the test performed? So the test is water holding capacity test. Number four, why is the volume of water collected so much and nothing is retained? Why is it like that? So the reasons for identifying specimen E is also the same answer here. So the soil is high in grit, presence of large stone, low surface area, and no cohesive bond holding it together. Number five, name one soil with high water holding capacity. Clayey soil. That is the answer. So clay has little pore, it has fine sand, it has binding, a cohesive binding force that holds it together. So clay soil has the capacity to hold water, and that's one of the reasons they use clay in making the old water containers that we use in those days, like the ikoko. They use the ikoko. They use them for making pots of soup and a jar of wine and so on and so forth. So, so number 6, name one plant that can grow on specimen E. Specimen E is sandy soil. Specimen E is sandy soil. So remember, so we have a variety of answers here. We have groundnut, cashew, cocoa, watermelon, and date palm. In fact, groundnut and coconut are uh, they are seeds or they are plants of choice that grow well on sandy soil. So we have specimen F, which is the alimentary canal of an animal showing the esophagus up to the stomach. Do not be deceived. This specimen is not a complete digestive tract. It starts from the esophagus, it terminates at the stomach. Well, you might also see a little form of duodenum. That's the little part of the small intestine, which is the duodenum. But there is no small intestine or large intestine or every other part. So that is one thing that you need to pay attention to. So identify specimen F, that is alimentary canal of an animal showing the esophagus up to the stomach. The next question, this is a common question. Between 8 to 10 or 10 to 12 centimeters long, draw and label specimen F. It might be the longitudinal section. In fact, this one is easy because the digestive system is like a tube. So it is the longitudinal section that you will be asked to draw. Many students fail to understand this question because this one has to do with the size of whatever you want to whatever you are asked to draw. So when I was in secondary school, I don't joke with diagrams because I know out of 10 marks, I'm going to score 7 minimum. So if I very lucky and I come across 3 diagrams in my questions, then I know I have 21 marks automatically because I was very good at diagrams. So here you are going to measure a box. WAEC will not penalize you if you leave the box or you clean it off at the end of the day. So you are going to use your ruler to measure 8 to 10 centimeters, or depending on what you are asked to do, 10 to 12 centimeters. What this means is that your diagram should not be smaller than 8 or 10, and it must not be bigger than 10 or 12 centimeters. So you are going to draw your diagram like this, and then you have the esophagus, you have the neck of the stomach above, that's where you have the pyloric sphincter, and then you have the stomach itself. Then the last curve, that is the junction between the stomach and the duodenum, which is the first part of the small intestine. The duodenum is directly attached to the stomach. So after labeling, you are going to label either to the right or to the left. It does not matter if your label goes out of the box that you have drawn. The only thing is that your diagram must cover 3 quarter out of 4. In fact, if I were you, I will make sure that my diagram covers from the head down to the bottom. It must not move out of that box. And I would suggest that you leave the box for safe reasons. So the first thing is you are going to write your title. For writing your title is just 1 mark. If you forget, you have lost 1 mark. That is that. Students tend to forget this many times when they are asked to draw any diagram. So the first thing is write your title saying diagram showing the alimentary canal of an animal from the esophagus to the stomach. That is your title. Then you label to the right. Make sure you use ruler. Your label must be what? Horizontal, and it must be ruled. It must be ruled. So that is that. And then after your diagram, because you are given the real object, you must find your magnification. Your magnification. Your magnification is always the image all over the actual object, I over A. Let's say your image is 10 centimeters and your object is also 10 centimeters. That will be M is equal to I over A is equal to 10 over 10. Or if your diagram is 5 and your object is 10, that will be your magnification will be 1 over 2, or your magnification will be 1. So for this diagram, your magnification or the expected magnification for this diagram is times 1 or times 1.5. Do not use decimal. Don't go and write mg is equal to 1.2 or 1.3. Please take note of this. Magnifications are always written in whole number or in fraction So you can say times, you will put times 1 times 1.5. That's 1 over 2 for this particular diagram. Let us continue. State the function of the following parts. So you have esophagus. The esophagus allows the passage of food from the mouth to the stomach through peristalsis. What is peristalsis? Peristalsis simply means a wave-like movement. A wave-like movement when food is being swallowed from your mouth down to the stomach through the esophagus. Then, what's the function of the stomach? It serves as a temporary food storage organ, and it also digests, turning the food into a semi-liquid solution or substance, a semi-liquid substance which is known as chyme or chyme, depending on what you want to call it. Number 4, which part of specimen F is HCl produced? HCl is hydrochloric acid, and state one function. So the stomach produces HCl. That's where it is produced from. The stomach is the one that produces HCl, and that HCl activates pepsinogen into pepsin, an enzyme which is responsible for the digestion of protein into peptone. So that's a partial digestion of protein into peptone. So here they can say, number 5, name 2 enzymes present in the stomach of specimen F, and state one function. Number 1 is renin. So it is an enzyme that curdles milk. It works on casein that is present inside milk and converts the milk into lactose. The second enzyme is pepsin, and it partially digests protein into peptone. Number 6, what is the suitable pH range for the enzymes to function? So for renin, the range is between 4 and 5, and the medium is acidic, or the pH is acidic. Then for pepsin, it's even more acidic, so it is between 1.5 and 2. So both enzymes work in acidic medium. Number 7, what is the type of nutrition the animal practiced? What is the type of nutrition? Because of the type of digestive tract that we have, so it is holozoic nutrition. It is animal nutrition. So An animal, according to their type of feeding, can be divided into three. We have the herbivores, we have the carnivores, and we have the omnivores. Number 8, state three functions of the stomach. Number 1 is it serves as temporary food storage. Number 2 is digestion of food. And number 3 is secretion of gastric juice. Number 9, describe the process of digestion from the mouth until the stomach. Number 1 is that food is ingested into the mouth, which is the buccal cavity. The teeth will masticate it, that is, the teeth will chew it into smaller molecules or into smaller particles. Then, from the mouth, salivary gland will mix with the food, and enzyme Canal will partially convert starch or carbohydrates into maltose, and then the tongue will roll the food into bolus, and then it will go through the esophagus in a peristaltic movement into the stomach. So the stomach activates the gland to secrete gastrin, that is the gastric gland to secrete gastrin, which produces HCl, and enzymes are activated to turn the food into a semi-solid substance called chyme. The next specimen we have here is a twig or branch of a plant with mistletoe, or a colored picture of a parasitic plant on its host. If I were you, I will not pick the first option because it may not be given. The picture might be given. So, and that's why they use the second option. So the second option is more likely to be used. So if I were you, I would, when they ask me to identify and give two reasons for my answer, so specimen G is what? A colored picture of a parasitic plant on its host plant. Period. No more, no less. Don't be looking for mistletoe or be looking for orange tree or kola nut tree or whatever tree. So just say a picture of a parasitic plant on its host plant. What are the reasons? Number 1, intertwined structure of the parasitic plant. That is, the ability to entangle with the host. Number 2 is the presence of historium. So number 2, what is the scientific name of specimen G? What is the scientific name of specimen G? So for the parasitic plant, depending on the type you are given, most times the most common one is mistletoe. The scientific name that we are going to give here is Viscum album. Remember that you are to start with capital letter and small letter, and then you underline them separately. If I were you, I will go with mistletoe. Number 3, what is the association that exists between the two plants? So the association is simply parasitism. Number 4, name 3 other associations that you know. We have saprophytism, symbiosis, commensalism, cannibalism, and so on and so forth. Number 5, they might ask you, define parasitism. Now, this is an association. You can see those are the keywords that you need to pay attention to. This is an association. It is an association between two organisms. This is an association between two organisms in which the parasite benefits and the host suffers harm. So the parasite is benefiting at the expense of the host. At the expense of the host. So number 6, name 2 methods to control the parasitic plant. 2 methods. Number 1, we have mechanical control by manually removing it. Number 2 is chemical control by spraying herbicides that kill the parasite and spares the host. Number 7, state 2 observable features of the parasitic plant to adapt on its host. So we have so many here. Possession of Historium for absorbing nutrients from the host, reduce scale like leaf to reduce the need for photosynthesis. Number 3, poorly developed root. Why? Because they already have historium that can get more nutrients from the host plant, so the roots are poorly developed. Direct attachment to the host, so they are directly attached to the host. So let's move on. Specimen H. Identify specimen H without reason. So a beaker containing milk. Number 2, put a small portion of specimen H into a test tube, add 1% of sodium hydroxide, that's NaOH, and add 1% of copper sulfate, that is, I usually call it Q-sulfur, and add Benedict solution to another portion. So you are going to get 2 portions of specimen H into 2 test tubes. One, you are going to add sodium hydroxide and Q-sulfur and copper sulfate. Then to the other, you are going to add Benedict solution. Record your observation and inference, like we used to do: test, observation, and inference. So the first one is specimen H plus sodium hydroxide plus copper sulfate. In your observation, a purple or violet or lilac coloration is formed, so indicating that in your inference, protein is present. Protein is present. Then the second one, specimen H plus Benedict solution plus heat, because you are going to heat it for 4 to 6 minutes. A brick red coloration is formed. So which means that we have reducing sugar is present. And when we talk about reducing sugar, you can say glucose is present, galactose is present, fructose is present, lactose is present, maltose is present. Sucrose is not a reducing sugar. Sucrose, I am saying it again, sucrose is not a reducing sugar. The reason is because it does not have an extra ring to react with the alkaline solution in Benedict reagent. So therefore, it will not be able to reduce the copper 2 into copper 1 oxide. So the inability of sucrose to reduce copper 2 into copper 1, it makes it a non-reducing sugar. So sucrose is a non-reducing sugar, while others are reducing sugar. So the simple sugars and the monosaccharides and the disaccharides, except for sucrose and trehalose. So Benedict solution can work. Phelline solution can also work for them. Number 3, what is the name of the test performed? The name of the test performed is Biuret test, and the second one is Benedict test. Benedict test. Number 4, name 2 other tests that can be performed for specimen H. For specimen H, we have Millon's test, that's for protein, xanthoprotic test for protein as well. Depending on the question, if they ask what other tests can be done, what other reagent can be used apart from Benedict solution, then you can also use Phelline solution. Number 5, state 2 nutritional values. So you have protein and calcium. Name 1 deficiency syndrome from the lack of specimen H, from the lack of protein or from the lack of calcium. So you are going to have 1, for the lack of protein, you are going to have kwashiorkor. This one is very, very important. Then the second one is rickets. That is, when somebody does not have enough calcium, it leads to weak bones, and then the bones might be bent like a bow. So we call it bow leg. We call it bow leg. So rickets. Now we are on specimen J. Specimen J is a fish. Identify specimen J with 2 reasons. So we have so many reasons here. Specimen J is just a fish. And then what are the reasons? Possession of fins. Do not repeat. I have come across students, when they want to give reasons, they will say possession of pelvic fin, possession of dorsal fin, possession of caudal fin, possession of anal fin, possession of pectoral fin. Please, all of these will be regarded as one. So don't repeat the fins individually. The possession of fins. The second one is possession of lateral line. Then you can also say possession of scales, possession of gill enclosed in operculum. You cannot see the gills because it is not observable from the outside unless you cut it open. So if I were you, I would say enclosed. The gill is enclosed in operculum, or I would just say presence of operculum. Another one is presence of compound eye or presence of terminal mouth. Presence of terminal mouth. Then the second one is classify specimen J into kingdom, phylum, and class. So like we used to do, our kingdom is Animalia, phylum is Chordata, subphylum is Vertebrata, and then class is Pisces or Osteichthyes. Now, if you want to pick Osteichthyes, you must be able to identify the fish you are given. If the fish is catfish or it is tilapia fish, then you can say Osteichthyes. But if it is not a bony, if it is cartilaginous, then stick with Pisces. WAEC will not penalize you for that. WAEC will not penalize you for that. But if the fish is tilapia or catfish, then you must add Osteichthyes. Then number 3, within 8 to 10 centimeters and 10 to 12, we have already addressed this. So label specimen, draw and label specimen J. So what comes first? You are going to use the largest scale, 10 centimeters or 12 centimeters. Make sure that your diagram covers from one end to the other end. Another thing is that make sure you use ruler to rule, and your label, your line of label must touch the part that you are labeling. Many students are fond of labeling irrationally. The label or the line of label will not touch the exact place where they want to label, and so the examiner will be confused which one is the dorsal fin, which one is the eye, or which one is the nose. Make sure that your line of label is touching the part that you want to label. And when you are labeling, start with capital letter. Make sure that your labels are ruled and they are not woolly. There is no shading. So specimen K, we have specimen K, and that is maize grain. So identify specimen K, name the type of fruit, and give one reason for your answer. Name the type of fruit and give one reason for your answer. K is a maize grain. And then the type of fruit, it is a Caryopsis, a simple fruit, but belongs to the Caryopsis. Then what is the reason for that? The reason is that the pericarp is fused to the seed coat. They are fused together, and so that is one of the reasons it is a grape. So number 2, classify K into kingdom, division, and class. So we have K is also a plant, just like the first rule. We have kingdom Plantae, Tracheophyta, Spermatophyta, Angiospermae, and Monocotyledone, because maize is a monocotyledonous plant. And so your final answer here will be kingdom Plantae, division Tracheophyta, class Angiospermae, and then you bracket Monocotyledone for safe reasons. Number 3, state 3 differences between K and A or B. A or B, we have already done that in some of the past questions. So 5 differences. We already have the 3 here. So the fruit type, one is Caryopsis, the other one is a legume. One is a monocot, the other one is a dicot. Then for endosperm is always present in maize grain. The endosperm. But in roasted or crushed groundnuts, there is nothing like mature seed. It has been what? It is only food storage. Then seed attachment. The seed is completely fused to the fruit wall, while seed is free inside the pod, is free from the pod. Number 4, state one similarity between K and A. Both are one-seeded fruit, or you can say they are both food reservoir. So let's move on to specimen L, tomato paste in a petri dish. So identify specimen L, we already said that. State the nutritional value of L. So we have vitamin A, vitamin C, iron, potassium, and dietary fiber. 3. What deficiency syndrome can result from the lack of specimen L? A major thing here is vitamin C, and that is scurvy, where the teeth and the gums of the teeth will be bleeding. So that is scurvy. Number 4, differentiate between L and M. L is tomato paste. So like our normal routine, we have our table. Seeds are absent in paste. Seeds are present. Processed into paste. It is fresh and raw. Low water content, high water content. No pericarp, mesocarp, or endocarp. Presence of pericarp, mesocarp, and endocarp. So we have specimen M, which is fresh tomato fruit. So identify specimen M. Then the reasons. What are the reasons? Presence of fleshy epicarp. Number 2 is presence of scars on both ends of the fruit. So if you understand fruit, you will find out that there are always two scars. The one is the remain of the style, while the second one is the remains of the receptacle. So that is why every fruit has two scars. Number 2, mention the type of specimen M. Type of fruit is berry. Why is that? Because it has multiple seeds. Multiple seeds. State the scientific name of specimen M. So the scientific name is Solanum lycopersicum. Solanum lycopersicum. The old name is actually Lycopersicum esculentus. Lycopersicum esculentus. So it has been renamed to Solanum lycopersicum. So if you write Lycopersicum esculentum, WAEC can give it to you. But the current name now is Solanum lycopersicum. Classify M into kingdom, division, and class. So you have class, kingdom, plantae, and so on and so forth. Then your final answer will be plantae, Tracheophyta, angiospermae. So it is a dicotyledonous plant. What is the placentation of M? What is the placentation of M? Remember, placentation can be defined as the arrangement of the ovules within the ovary of a plant. So the name of placentation in M, the placentation of M is exi-placentation. Exi-placentation. I'm going to explain this when we have the picture. Number 6, state one nutritional value of M. Vitamin C, I have already said that. Vitamin C, potassium, vitamin K, folate, and lycopene. Number 7, state 5 observable differences between M and N. So M is fresh tomato fruit, N is rotten tomato fruit. So number 1 is M, bright red color with no patches. The other one is brown or black color with patches. Then the second one is the fresh tomato is fresh, smooth, and the skin is intact, while the second one is broken or have ruptured skin. Then the first one is odorless, while the second one has foul smell or putrid smell. Putrid smell. Next one is it is firm and turgid. It is firm and turgid. But the second one is the rotten tomato is soft, mushy, and liquefied. Then for fresh tomato, the seed is intact, while seeds are partly decomposed. Seeds are partly decomposed. Another thing that might come up is that they can say that you should test tomato, the rotten tomato, that's specimen N, with Benedict solution. You might test it with Benedict solution, and what you are going to have is a negative result. Why? Because the glucose inside tomato has been used up by the bacteria that is decomposing the rotten tomato. Number 8, cut the transverse section of M and draw between 8 and 10 centimeters. We already explained this. So here I wanted to talk about placentation the other time of tomato. So the placentation we are looking at here is axile. And axile placentation is similar to parietal placentation. For parietal, there are 3 walls coming out from the margin, and then the 3 walls are facing each other, and then the seeds are orientated towards the center of the fruit. But this axile in tomato, we have the orientation of the seed. It's like there is a margin, the walls are coming from the center, and then we have a segment, and the seeds are orientated towards the margin or towards the wall of the fruit. So you have tomato, which is axile. Another one that is like this is orange. So both of them have axile placentation. So N is rotten tomato fruit. Identify and give one reason. So the reason here is visible sign of microbial activity or decomposition. Number 2 is discoloration or loss of structural integrity. Number 3 is foul odor and presence of mold. Name 2 organisms responsible for the condition of N. We have bacteria and fungi. What phenomenon does N illustrate? What phenomenon? So the phenomenon here is diffusion. In what state is this diffusion? It is gaseous state. So because the smell of the tomato, of the rotten tomato, will diffuse into the air, and people will be smelling the bad odor. So number 4, state 4 economic importance of the organisms responsible for N. So number 1 is that the microorganisms, either bacteria or fungi, cause spoilage of the fruit. They reduce the market value of tomato. They produce mycotoxins or toxins. If it is for fungi, it is called mycotoxin. If it is for bacteria, we can say bactericin or bacteria toxin, leading to food poisoning. If you consume anything like that, any spoiled food, then it can lead to food poisoning. Then it increases production cost because the farmer will want to spend more on procuring some other facilities to ensure that the tomato is well preserved. Then number 5 is decomposition of dead matter. This one is not related to the plant, to the crop. So of dead matter, generally microorganisms are decomposers, and they are responsible for the decomposition of dead organic matters, and that is nutrient cycling. So number 5, state one method of preventing N from decay or rot. So number 1, you refrigerate at 10 to 10 degrees centigrade to slow the growth of microorganisms. You can can it, you can bottle it, you have freezing method, you have preservatives, you have salting, you have pasteurization. Then, so you can just choose any one depending on the question you are asked. Depending on the question you are asked. Specimen P is thermometer, Q is meter rule, and R is quadrat. R is quadrat. Identify P, Q, and R without reason. Without reason. So thermometer, meter rule, and quadrat. State one function for P, Q, and R. Thermometer measures temperature of a body or substance. Can be, if it is not a body, it will be air, soil, water. For meter rule, it measures the length or distance up to 1 meter because that's the maximum length of any meter rule. The quadrat, that is for R, it delimits a sample area. That is, it reduces the area so that you will be able to count the number of organisms within that area. So it's like a mini habitat. It creates a mini habitat. So it delimits a sample area for ecological studies like estimating plant population density, frequency, and coverage. Number 3, state 2 precautions for using P, Q, and R. So if you want to use thermometer, number 1 is handle carefully to avoid breakage. Number 2, do not exceed the temperature range that is marked on the thermometer to prevent damage. So for meter rule, avoid parallax error by reading the measurement with eye directly perpendicular to the scale. Number 2, keep the ruler or the meter rule dry and straight. Do not use a bent or warped meter rule for accurate measurement. Then for specimen R, place the quadrat randomly or you throw it randomly to avoid sample bias. Then number 2, ensure that the quadrat lies flat on the grass without gaps and count only organisms fully or partially within according to fixed rule. That is, include those touching the top and right edges. So thank you very much. If you like what I do these 4 things for me. For more videos like this, subscribe, like, comment, and share. I would like to hear from you, or you want to book a coaching session with us. Do not hesitate to put it in the comment. Thank you very much. See you later.